people in the panel have spoken today, so I'm not going to introduce them. I might only want to introduce Aubrey de Grey, who hasn't spoken. He's the Chief Science Officer of the SENS Foundation. And then next to him is Edouard Debonneuil from HEALS and the International Longevity Alliance. And the other panel members have spoken, so you're familiar with them. As you know, we are going to talk about the hallmarks of aging. And just for those who might not be familiar with them, I put a slide up behind you with the 9 hallmarks of aging. So maybe I can go over them. There is DNA damage, there is senescence, we have had lectures today on that topic, there is stem cell depletion, there are epigenetic changes which were also mentioned today, mitochondrial dysfunction which was briefly touched upon as well, deregulated nutrient sensing, telomere shortening which was also mentioned, loss of proteostasis which we just talked about, and cellular communication changes between different cells. Okay, I would like to start by asking the panel members if you think that having a classification system like this has its use in clarifying the thinking about a complex process like aging. And I know Aubrey has actually proposed another mechanism and a set of 7 characteristics which are quite similar actually to the 9 ones. So I will like to hear his opinion on that.
When I saw this paper, I thought it was a fairly good approximation to SENS, really. Just the fact that it was 10 years late. Of course, some of the things are identical to the 7 things that we have been talking about since the year 2000. And the ones that aren't are, I think, questionable only in the sense that they are not driven by being tied to particular proposed therapies. The real motivation and inspiration for the 7-point plan of SENS is precisely that within each of the categories there is some kind of generic therapy, either a therapy that pretty much already exists in some cases, or at least one that we can describe in quite a lot of detail, and which ought to work across the board for every example within the category. So that's the main reason, that's the main thing I think is not entirely right about this classification, but I was very pleased to see that that such distinguished authors were able to put together something like this and publish it in a nice prominent place, and maybe it will gain a little more traction now among the mainstream than it has had before.
I think that modeling, that's the way to model the aging process, is a good way to find out what is and what isn't in this model, because no model is perfect. Maybe I will just, for example, indicate that I wonder where to put thymic involution. So maybe it's easy, not to me. Just an example, of course. And of course we...
I think that's an easy one. That's basically just stem cell depletion. There's cell loss, not... And that's in both SENS and this.
Thymic involution is a developmental process. It's nothing to do with stem cell. Yes, it is.
No, it's not.
Of course it is.
No, it's not.
Well, thymic involution occurs in, as far as one knows, essentially every animal that's been looked at a certain time in their developmental progress, way before any signs of the aging version of senescence, rather than the developmental version of senescence, where cells accumulate with markers that are seen in senescent cells, but which do not show, as you said, you don't like the word senescence, so one shouldn't call those cells senescent. Senescence means something to do with aging, which is degenerate. That's the meaning of the word senescence. So if you want to call it senescence, you have to show that that is the case. Whereas thymic involution and developmental aspects of senescent cells are developmental processes that do not directly contribute to the aging process, because where development stops, aging starts.
Just to say, this is exactly what I was saying. The fact to put precise things where in fact it's a combination or a question of what we mean behind the words allows us to better decipher how we think aging, and in fact to ask questions differently than if we were a bit not very precise. So it forces us to ask such questions.
I guess I just wanted to comment. I think that aging is an extension of development. I mean, I really think that we place names on these time periods, and I think it's all part of the same process, actually. So I think it's difficult to separate them into different processes. Go ahead, hit that. No, but the other thing I wanted to mention about these hallmarks is that they're all very interconnected. I think you don't see one without the other in a lot of animal systems anyway. That some that have mitochondrial dysregulation also have nutrient sense and dysregulation and that type of thing, and a loss of proteostasis. So I think they're all very interconnected. It's hard to single out one or the other.
Well, I think the difference between development and aging is that for development there is a clear program. And with a clear program, I mean that that an embryo develops the structures component this organism at specific stages. So there is a genetic program specifically at specific stages to be developed. But in the case of aging, every single person ages in a different way from the rest of the people. So I think this is the difference. I don't think there is a real program for aging. And I think dividing the hallmarks of aging in categories may be very useful. We should also determine which of these hallmarks of aging are primary hallmarks, for example, telomere attrition or genomic instability or epigenetic variations or the loss of proteostasis are primary hallmarks because are always detrimental. However, there are antagonistic hallmarks, for example, cell senescence or the deregulation of the nutrient sense, which are beneficial at low levels, but at high levels can be detrimental. So I think this is very, very useful to have everything divided into categories, but it is even more challenging to combine and to integrate all this knowledge. And I think to integrate this knowledge is the future for therapies to extend lifespan. So this is really challenging.
In fact, we will learn as we will learn. So for now we have views, and so it's not a matter of modeling. I think we all agree that here it's more a matter of deciphering what are the most events that come with aging, and we will base a hypothesis on that, and we will make some tests. So perhaps, for example, tomorrow we will discuss mouse lifespan tests, and perhaps it will become quite useful, quite general to test the different hallmarks on mice and discover that in some cases it goes that way, the other way. Like today we've seen that ages can be good to eat in order to prevent cancers. So things we will see as we learn, but we need some markers and some commonalities in order to speak the same language and advance together and build upon each other.
Okay, let's go to the second question. Although we already touched a little bit on it, do you agree with the proposed hallmarks? And do you think there is a possibility to discover yet unknown hallmarks of aging?
So I think most of these hallmarks are uncontroversial. I think that one could have come up with them 20 years ago, in fact. But I think that if we ask, is this a complete list, then we have to look at it very closely. Certainly, Science Research Foundation has been doing that for the past 15 years. And I'm pretty happy that the 7-point plan that we put forward a long time ago now seems to be standing the test of time. I can point to things, even though I think thalamic involution isn't one of them, that I think are missing from here, like macular degeneration, for example, the accumulation of molecular waste products that are not proteins is not mentioned here. But there's not much wrong with this. No, I think it's fine.
And also perhaps, but perhaps that might be the next question, I guess. It's one thing to understand biology, to put some words and to work together and to discuss them. It's another thing to be able to bring the whole world with us, and in particular the pharmaceutical industry. If people can view aging as a set of diseases or processes that are identified and start work with that, I think that eases the process. Because otherwise aging is something a bit magical that we don't know how to appreciate. And here it gives us very specific clues and so ways to think and to develop some therapies and funding and processes.
Well, I want to open the possibility to other novel hallmarks of senescence, of aging, sorry. But I think these are the main ones. But I think the main problem, as I have said, is to integrate all the information, which is huge now.
Well, the question was what's missing, if anything. Looking at it from my own parochial point of view, as I always do, of course, I remember that this scheme is based on the hallmarks of cancer that had appeared in its original version in 2001 or so, and that missed out on immunology. And the new version from a year or so ago of Hallmarks of Cancer has got immunology writ large. This one hasn't got any immunology directly, of course. All of those mechanisms, or most all of those mechanisms will be applied to immunity, but we go back to 1967, and Roy Walford, the previous speaker, mentioned this interesting pathologist working on tissue typing, but also very interested in aging, plus the fact he was a friend of Timothy Leary and other interesting people in San Francisco at that time. And he wrote in 1967 a book called The Immunologic Theory of Aging. A long time ago, he thought that dysregulated immunity and the mechanism being the generation of autoantibodies was responsible, or he proposed this was responsible for the main diseases associated with aging and for much of the aging process itself, because you are attacking your own tissues with antibodies. And I would say that this should be on here now in the version which doesn't only involve autoimmunity via antibodies, but a dysregulated immunity which occurs for via many of the mechanisms that are mentioned here, but with the readout, so to say, in terms of the extremely dangerous effects that immunity has, in that an acute immune response that will protect you against a pathogen also has a great deal of tissue damage associated with it. So all the symptoms you get when you get flu are immune pathological, not caused by the virus. And if we extend this into a chronic situation over 80, 90, 100 years, then you are getting an accumulation of damage mediated by this, as age progresses, less well, for whatever reason, some of these events being the reason, less well regulated immune responses that I think deserve a place. Which colour could they have? We find a different colour for immunity there, bright red maybe, and put that onto the hallmarks of aging as well.
I agree with that. I think these types of publications that point out the hallmarks, they're not complete yet, they need immunology, I agree with that, are really important because it gives people targets to go after. And I think the problem is, the reason this is important too for targets is because, at least in the United States, the FDA is not going to approve a drug for aging. They need to approve a drug for something specific on this list somewhere, or another list that's generated that is age-related involves age-related processes or disease targets, because they're just not going to get any funding or any work to move forward with research and development into something that just, stops aging or reverses aging or slows it down.
That's the unfortunate position in the US. Perhaps some places in Europe are a bit more.
Now there's an increasing interest in cell competition, for example, which is another process which was discovered in Drosophila, by which a winning cell can mediate the elimination of the loser cell. And it has been related to cancer, for example. And I was wondering also whether this process may have a role during aging. So there are many possibilities, but there is an emergent interest in this process, in cell competition.
Also behind, after this 9 or 10 or more hallmarks, it would be nice to become more precise. For example, to agree, even if not everyone will agree, it will be a consensus on some things to test, some markers, so that we can really build upon each other and become a lot more concrete. Even if with time we will discover that something is lacking, but I think it's because we will define some first definitions that then we will more easily find that something is lacking.
Yeah, I was very surprised that Holly didn't complain about the absence of endocrine aging up there, which seems to be on the face of it just as egregiously absent as immune aging. But what it reminded me of was that in the very early days when I was first describing SAND back in 02 and 03, if you go back to those papers, you'll find that there are 9 points in my classification, not 7, and the 2 additional ones are endocrine aging and immune aging. And the reason I eventually started checking them out was because I felt that they could be described more accurately and more usefully as falling within the other categories. So immune aging being loss of cells in the case of thymic involution, and accumulation of cells that wouldn't die in the case of CMV-specific effector memory cells. And with endocrine aging, similarly, either loss of number or changes in the internal structure of cells in respective glands or in the cells that respond to hormones. So again, this classification isn't all that different from what we've been thinking for quite a long time.
Okay. So what are the implications, if any, of the hallmarks of aging on translational medicine?
Someone else go first this time.
Well, at least in terms of drugs, what I just mentioned a second ago with the FDA, at least in the US, that I think these provide targets or pathways for people to go after and molecularly primarily, and then some potential antibodies that can be made to some of the different things in the pathways to either slow or stop some of these processes from occurring. So I think that you could slow down some of the processes with targets, but I think at least there may be like more liberal advantages to working outside the U.S. in terms of targets, because it's just the aging is just not going to be a good target to go after, or to promote targets, at least in the pharmaceutical industry in the U.S.
Yeah, that's probably true in Europe as well. It's nothing to do with liberal or illiberal. I think it's to do with indeed the selection of a target which appears to be amenable to manipulation. Although these are more basic science targets, so the question of how does this really input into translational medicine, I would say at the moment probably, yeah, the question was does it at all? And at the moment I would think probably it's more likely not to at all.
Well, I think that to extend lifespan, probably we will need to target different hallmarks, not only one, because this is a really very complicated.
Anybody's tried to do that practically in translational medicine? I don't think so.
Yeah, it's difficult. But for example, you were talking before about the immune system. In the case of senescence, the problem, the detrimental part of senescence, because I think that it is intrinsically a beneficial response, because it is aimed to eliminate damaged cells or unwanted cells, as it is the case as development. So in the case of senescence, the detrimental part is because senescent cells are not removed, and when these sequential steps are not completed, senescent cells accumulate, and due to the SASP generate a pro-inflammatory phenotype. And in this case, I think that, for example, the immune system is very important during aging, because an inefficient immune system can generate also detrimental parts of these hallmarks, for example, in the case of cell senescence.
So about translational medicine to humans, there is the agreement by the regulator, but there is also the feel that something is needed by the client or the person. And so here, for now, we have only beginnings of regular testings. For example, with DNA SNP testings like 23andMe, more and more people start to look at it and see that they have something specific or not. And here, if I look at telomere shortening, it starts to be a small business to test that and to test it in many ways so that it becomes more and more accurate as knowledge comes along the way. Same for genetic changes, but that's a bit further away. And same for DNA damage, but that's also quite far from today. So I would say that this is it seems to me this is a good roadmap to go to medicines and perhaps to humans in perhaps 20 years, because we will need to have the time for those tests to come, for then people to realize that perhaps this is very interesting to them.
Yeah, I think that's right. I'm actually not too worried about the regulation step. When I talk to people from the pharmaceutical industry or from the medical industry in general, I get the impression that if they think something's going to sell, they will find a way to get it approved. Even if it involves, first of all, saying that it's for such and such a disease and then, spreading its application elsewhere later on. So I think the difficulty is, as you say, with the public, with the client, that we have two big challenges with this kind of medicine. Number one, that by the nature of aging, as we see with the hallmarks here, it's going to be a combinatorial therapy, which is something that people are scared of. And secondly, that it's going to be a preventative therapy, which is also something that's generally a hard sell.
Okay, thank you. Can the hallmarks, some or all of the 9, be used as a biomarker of aging in humans? And that's what Edouard was touching upon. So can you basically measure telomere shortening, mitochondrial dysfunction and so on, and put them together in some type of model where you will then, give them some type of, how biologically old a person essentially is and what he can expect?
Yeah. I kind of think that's the wrong question, because on the one hand, these things, yes, they can be measured in some cases with current technology, like we can measure telomere shortening in lymphocytes. It's a good deal harder to measure telomere shortening in a lot of other tissues, and the same is true for, I think, probably all the examples up there. However, the great news is that all of these things happen in all of us. They happen at different rates in different people, but not very different, really, if you think about it. There's a reason why hardly anyone dies of the diseases of old age before about 60, and everyone does so before 120. It's pretty good news that we're all aging at roughly the same rate. So it may end up being the case that as and when therapies like this come along, it's more efficient simply to provide them a bit more often than necessary, rather than to engage in any kind of diagnosis.
So I am not able to know if we age at the same rate for the 9 hallmarks yet. Today, this morning, we were seeing that the elasticity of some tissues was probably the thing that was the most striking at early ages, whereas the neuron degeneration, basal degeneration, was probably the latest. Maybe the word isn't exact. We will learn with this, and whether it's the same for all or if it's different for all. I think in both cases it leads to pharmaceutical or approaches, because if we start to be a patchwork of aging persons in different ways, we will perceive it as a personal disease that we have or a condition, and we will want to treat that specifically. So it will depend on the results, but it gives us clues to really advance forward.
Once again, I think that it is difficult to obtain a unified biomarker of aging, and aging should be defined by different biomarkers, in my opinion, of all these hallmarks, involving all of these hallmarks. For example, in the case of senescence, it appears that there is an accumulation of senescent cells during aging, which are positive for the cell cycle inhibitor p16. And in other hallmarks, for example, in telomere attrition, there is an evident shortening of the telomeres during life. And in the laboratory of Maria Blasco at the Spanish National Cancer Research Center, they are working hard on this, and they have she has a company to measure the length of the telomeres for the people as a potential biomarker, and preliminary data. They have determined that apparently there is a correlation between age-related diseases and this telomere shortening, but they are obtaining all the data now. They are making a database for many people, and is what happened in these cases, in telomere attrition and in cell senescence.
Yeah, well, it's a very tricky question. I think the answer is yes, biomarkers, certainly we do need them. We would like to have them, but it's easier to say than it is to do. We need big data, big in terms of the number of different people that are being studied, because humans are so heterogeneous, even of the same race. And when we look outside of that, with a genetic background different, environment different, we have to do individualized medicine, as it were, or individualized studies, but on a very large scale with a large number of people. It's no good looking at animal models. You've got to look at people, you've got to do longitudinal studies of people. If you have a biomarker which simply associates at young age with a certain outcome, at old age you see that the biomarker that's present in the young is not present in the old, so it must have been bad for you because old people don't have it, they all died beforehand, and vice versa. This is not really going to tell you anything really important. You have to have, the biomarkers are only useful if you know that you have a robust clinical correlate, even without causality. You have to have a robust clinical correlate. Mortality is the most robust one. What else are you going to take? You know, age-associated disease, cardiac disease, cancer, response to vaccination. It's all so heterogeneous in people. You can't take this as an endpoint and say we have a biomarker of aging which correlates, then predicts this age-associated disease. So one of those targets there is the one, and that's the one we should look at. I'm sure it's not going to be that simple, and that's already not simple. So that we need more data before one can really do anything useful. I think if you found one marker like telomere lengths in leukocytes, I think it is, isn't it, rather than lymphocytes, that's the big difference, but still, yeah, it's good to go for one, but it's not good to say that really this is a product that we can promulgate as giving an individual person some meaningful information about their health and aging trajectory. I think it's a very bad idea to do things like that. We still need to keep this entirely, I believe, at the academic level and do much more expensive studies.
I guess the only thing that I have to add, I mean, I agree with Aubrey about the rate of aging in humans is probably similar, but if you break it down by age-related disease, then the rates are very different amongst people, and that has to do with the heterogeneity, I think. So it's kind of the total package type issue that we're looking at, which is going to be very, very difficult to predict how long people are going to live, and it's dependent upon your genotype and your environment. So I agree that this is a good start. It provides targets, again, for us to look at, but everyone's not going to fit into one single category or all of them together. I think it's going to take huge numbers of people.
Huge amounts of money and time.
Yes, but similarly that we have a blood test to determine the quality of our health, we could have also an aging test with different parameters. We should need many of them, but it could give us an idea of the state of aging of that person, in my opinion.
But do you want to know?
No. Exactly.
Yes,
This is challenging, of course.
Yes, because you want it to be translational, so that when
So-
you know you can do something.
Exactly. But what I mean is that we will need many different biomarkers, not a unified one.
Just as an example, I have recently read a description of a methodology for a demographic study following a healthy longevity and so various markers, not biology markers, but more health style, in order to visualize how a population evolves with age depending on their actions in life. And they typically need, well, they have made some calculations based on the heterogeneity of what people do in life. And if they have less than 3,000 persons, it's not worth looking at what explains what. So this means that we will, in order to have a good vision of what is useful for health, for long-term health and longevity, we will probably need to have some cohorts that have at least a few thousand persons that are identified like that.
Okay, let's go to the last question, and then we end with just a few minutes delay, so very good done. So what do you think of these 9 hallmarks of aging? Is the one where we can most quickly have some type of experimental therapy that can be tested, for example in mice even, and show some result on lifespan extension. So we heard about Brichos, Gosling breakers, telomerase today, and several other things. So what do you think?
Well, I think most of these can be tested in the lab, in mice, so long as the correct mouse model is used. We've obviously seen already cases where mouse models that have been arranged to have an accelerated accumulation of one or another of these hallmarks can then have a rejuvenation therapy done to them that eliminates that accelerated damage. I'm thinking especially of the case of senescent cells with B16, but there are other cases. So I think that's straightforward. The question is whether this has, how much relevance it has to taking things forward to the clinic. Because of course in the clinic we really do, as has been said, have to do all of these things at the same time. However, saying that, we have to be very optimistic. I think actually the case of the B16 positive cells is a wonderful one because that was only a result in mice. It was a very preliminary result really because it was a genetic accelerated aging model and therefore it gave no clues for how to develop a drug that would eliminate senescent cells in humans. And even the longevity wasn't actually increased because of the heart difficulties that these mice had. And yet, the people who did the study went out and formed a startup company and they were successful in bringing in a very respectable amount of seed funding, I think it was around $7 million, to take it forward into the clinic, to actually do a search for drugs that would do this. So this really says that maybe there is an increasing understanding on the part of at least some visionary investors that an anti-aging industry built on products that actually work is going to be unprecedentedly enormous, and therefore that it's worth actually taking bigger risks than one might normally take as an investor.
I would like to mention that actually I heard a few days ago that an update on that paper will be published in a few months. So we are looking forward to hear that.
And also going forward and also backward with what we've said about the numbers. Actually, there is one big question, which is the transferability of mouse results to humans. And one way to potentially address it is to, but that's a limited way, it's to start from humans. So it's today difficult to analyze if, for example, the drugs we take, what we do in life, impact our mortality because we live long, so it's just difficult to, even if it's not long enough, we still live long, and so to get the data. But if we start to have those data in humans, we will start to see what things do in a relatively short period of time, and this then we can look at it in mice with the translated markers in mice, and we can see if it also works in mice. And if it does, then probably it works in humans because it's coming from humans and then tested in mice. That might a lot increase the translatability to humans because we will have those markers. Otherwise, in humans, it's difficult to measure things.
Well, in the case of mice, I think that all of these hallmarks have been studied and are in a continuing research, experimental research. But I think that what is challenging is to combine different hallmarks in the same mouse model. And I think this is a very important point, because at the end we all suffer the combination of all these hallmarks. But in the case of human, I think that I'm very positive. I think we are really far away from the top limit of years that we can live. And I think that we have many things to do in a near future to extend our life extension.
Yeah, we need to gather lots of data from humans on all of these hallmarks, but the question was which one would we go for? I would vote for DNA repair.
I'm going to disagree with you.
You vote for something else.
No, I would go for dysregulated nutrient sensing. I think that's something that can be targeted relatively easily and sort of is through diabetic medications and things right now are seeming to have some more impactful on other metabolic systems within humans for sure. And in mice, I think, like the other panelists have said, a lot of these already apply, and there are some models that integrate many of these. My own bias with the dwarf has changed. Their mitochondrial function is increased, and nutrient sensing is dysregulated just a little bit anyway. But it has has some of these same hallmarks that we've already found. So there are models out there that have multiple hallmarks of slowed aging or delayed aging. But again, so for humans, I think the dysregulated nutrient sensing would be the one to go after. That would be my favorite one.
Okay, and tomorrow I will give a lecture where I will actually use the 9 hallmarks of aging to look at different geoprotective drugs. So that's going to be interesting. So this is the end of today. I would like to thank the panel. I would like to thank all the speakers, the people who attended the conference, and of course the sponsors who sponsored this event, the Sense Foundation, Longevity, the Forever Foundation, and International Anti-Aging Systems. Thank you very much.